lapel.
Welcome back to our channel. In today's video, we're going to dive into the fascinating world of genetic engineering and explore the methods used to understand genes, genomes, and other omes. So, grab a cup of coffee, and let's get started. Genetic engineering has revolutionized the study of gene organization, structure, function, and expression. However, once a gene is isolated and sequenced, there's still much work to be done to interpret its various structural features in the context of its function in vivo. To better understand genes, scientists search for regions of interest such as promoters, enhancers, and protein coding sequences. While bioinformatics plays a crucial role, experiments are necessary to determine the location of these important gene regions. One method used is gel retardation. A restriction map of the cloned DNA is prepared, and a set of restriction fragments is generated. The protein under investigation is added and allowed to bind to its site. The fragments are then subjected to electrophoresis. The DNA/protein hybrid will run more slowly than a control fragment without protein, and its reduced mobility helps identify the location of particular binding sites on DNA molecules. Another technique called DNA footprinting is used. A DNA molecule is labeled at one end, and the suspected regulatory protein is added and allowed to bind to its site. An untreated reaction without protein is also set up. DNase is used to cleave the DNA strand under specific conditions. The region protected by the bound protein will not be digested. By comparing the treated and untreated reactions, the position of the protein on the DNA can be determined by its footprint. The chip assay involves cross-linking the protein and DNA using formaldehyde. The unbound DNA is then sheared into fragments. Specific proteins of interest can be precipitated using antibodies, and the associated DNA sequence can be determined. This method helps identify the protein-DNA interactions in a genome. Locating the transcriptional start site, TC start site, is crucial for understanding gene expression. Two methods commonly used are primer extension and S1 mapping. In primer extension, a labeled primer is constructed that hybridizes near the 5' end of the target mRNA. The size of the cDNA fragment is determined by electrophoresis, providing information about the location of the TC start site. Gene expression can be studied using rDNA through two main methods. Firstly, genes that have been isolated and characterized can be modified to study the effects of the modification. Secondly, probes obtained from cloned sequences can be used to determine the level of mRNA for a particular protein under various conditions. DNA microarrays, also known as DNA chips, have revolutionized gene expression studies. These microarrays exploit the base pairing features of complementary nucleic acid sequences. By immobilizing a large number of different sequences on a glass microscope slide, researchers can hybridize them with a fluorescently labeled sample. The binding of complementary sequences can be analyzed using laser scanners, allowing us to study gene expression on a genome scale. Mapping genomes is crucial for understanding the organization and structure of genes. There are two main methods, the shotgun method and the ordered strategy. The shotgun method involves sequencing random fragments and assembling them using sequence overlaps. The ordered strategy involves genetic mapping, physical mapping, and sequence analysis. Genetic mapping relies on the analysis of recombination frequency during meiosis. By studying the frequency of recombination between genetic markers, the relative position of genes on the chromosome can be mapped. This method helps us understand the linkage between genes. Physical mapping involves the construction of a map that shows the specific location of markers on the DNA sequence. Clone mapping, fluorescent in situ hybridization, FISH, long-range restriction mapping, radiation hybrid mapping, sequence-tagged site, STS, mapping, and expressed sequence tag, EST, mapping are some of the methods used for physical mapping. Understanding genes, genomes, and other omes is a complex yet fascinating field of study. Through various methods like gel retardation, DNA footprinting, chip assay, and DNA microarrays, scientists can unravel the mysteries of gene structure and function. Additionally, genome mapping techniques provide valuable insights into the organization and arrangement of genes within a genome. That's it for today's video. We hope you enjoyed this deep dive into genetic engineering. If you found this information interesting, don't forget to hit that like button and subscribe to our channel for more exciting content. Thanks for watching, and we'll see you in the next video.
Pharma Pal.